Dr. Joe, he's from the biology department of the University of California, Riverside. His lab uses legume rhizobium symbiosis system to study the plant microbe mutualism for years, and has made a whole great contribution to understand in the field to understand the mechanisms of bacterial mutualist explanation, host defense, and how this system involved. Hi, Dr. Sachs. Take your turn, please.
Well, thanks so much. So as in the introduction, in my lab, we study symbiosis. And I'm going to be talking about quality control between species. But what is really nice, and I thank all the previous speakers for their really nice work, is you're going to be able to see some integration that is occurring at these different levels, from the levels of cells to organs, and potentially even down to the level of the genes. So one of the most striking aspects of symbiosis, or these intimate interactions between hosts and their microbes, is the recurrent and diverse evolution of symbiotic organs. Now, we can see some symbiotic organs in this picture here. And what symbiotic organs are host structures that have a suite of three important features. One, they house very dense populations of microbial symbionts. Two, these organs, their development is induced or is significantly altered only in the presence of those symbionts. And finally, the phenotype of these organs is an emergent feature that is shaped both by the genome of the host and by the symbiont. Okay, so what we see are hosts that have evolved these complex organs in order to house symbionts. And of course, this is driven by selection. They're gaining benefits from the symbiotic services. But what's interesting is that the host becomes vulnerable when it develops an organ in which it's giving over a little bit of genetic control to the microbes. So we want to study what are some of the repercussions of the host giving up control. Can that lead to exploitation by the symbionts? And then we'll turn the system over and try to understand how can the host maintain quality control in this kind of system. In my lab, we study symbiotic organs that have evolved multiple times within the legumes. And of course, this is the legume rhizobium symbiosis. And these are the two questions that I'll be addressing in this talk. Do symbionts exploit their hosts within these symbiotic organs? And to what degree can hosts defend themselves against this exploitation? All right, so this is an association that can be described as a mutualism. A mutualism is an interaction between two different species in which each is getting a significant fitness reward from the interaction. Of course, rhizobia, they're going to form these nodules on the roots of legumes within which they fix a significant amount of nitrogen for the host. This expands the host niche and allows it to grow much more rapidly than it would otherwise. In turn, the host is going to house these rhizobia intracellularly within the plant cells and give them essentially a continuous supply of carbon from photosynthesis. So each partner is getting a big reward from this association. But over the long term, biologists, evolutionary biologists in particular, have predicted that this kind of system can be vulnerable to exploitation. And we'll focus on exploitation by the symbionts. One main reason is that the service that the rhizobia are providing to host, nitrogen fixation, is extremely costly. Every time they break that triple bond in a dinitrogen, it's going to cost them between 16 and 18 ATP. So if they could stop that service, they could gain a big metabolic advantage. The other reason we predict cheating from the rhizobia or exploitation is that they have a lot of molecular space that they can explore. The microbes in any of these associations have a very short generation time compared to hosts, and they have a very large population size. So they're always generating new mutants that can potentially evolve to exploit the host before the host has evolved in response. So in this system, we're going to be looking for a particular kind of cheater phenotype, and this would be a non-fixing rhizobial strain. So to summarize, this is a strain that can form a nodule on a host, and thus it's gaining benefits from the host, but it's not going to be fixing nitrogen in exchange. All right, so first let's address that question. Do symbionts explore their hosts? To test this question, we decided to first study it in a natural population. And what we wanted to do was try to understand the degree to which this cheating phenotype evolved in systems that were unmanaged, that were not affected by human intervention. We decided to study a relatively widespread plant species, Acma spongijosus. This is a small leguminous herb that can be found throughout the American Southwest in arid and semi-arid habitats. We focused on a metapopulation of these plants that range from southern to northern California over about 1,000-kilometer transect. So we studied many populations. Whenever we found plants, we would dig them up and we'd culture all the rhizobia from each of the root nodules. We'd archive those strains and genotype them, initially at 4 or 8 genes, and ultimately now we sequence them for the whole genome. We'd reconstruct phylogenetic relationships among those strains, and then we'd be able to start building detailed epidemiological maps of these populations across the state. A couple important facts from these data. One is when a host digs its roots into the soil, for the most part, each nodule is going to harbor genetically unique rhizobia. There's a lot of diversity out there. But on top of that diversity, we occasionally find what we describe as epidemic strains. Just like a pathogen might have a very successful genotype that spreads across populations, some of the same processes can also happen in mutualistic microbes. All right, but what we are now focusing on are the phenotypes. And what we want to understand is how often do we find exploitative strains that fit the description that I described as before, ones that form nodules but fail to fix any nitrogen. We decided to survey isolates from 10 different populations across the range, covering about a 750-kilometer transect. For each of these isolates, we grew them up and we inoculated them onto multiple diverse host genotypes that had been developed as inbred lines of Acma spongijosus. When we conducted these inoculation experiments, we found that about 7% of those isolates could be categorized as non-fixing. So they formed successful nodules, but they did not fix any nitrogen at all, and the host did not grow when they were inoculated with these strains. All right, so these non-fixing strains appear to be relatively common. Now we want to try to understand their evolutionary origins. So we're going to reconstruct this trait within a phylogenetic tree. So there's multiple ways that you can reconstruct a tree with these bacteria. They have what we describe as a bipartite genome, including a chromosome in which most of the genome is encoded, as well as a small symbiosis island. This is an integrative conjugative element that is similar to a plasmid that can integrate into the genome and then pop out and be horizontally transferred. For now, we're just going to reconstruct a tree using the chromosome, but I can also share the data with the symbiosis island, which is similar. When we map the trait of nitrogen fixation onto the tree and studied the losses of nitrogen fixation, we can infer that there have been between 4 to 6 independent losses of nitrogen fixation from an ancestral case of being mutualistic, fixing nitrogen for the host. So we can see a couple key patterns from this. One is that the non-fixing strains are not really related to each other, so there's multiple origins. And in every case, the non-fixing strains are very closely related to taxa that are beneficial. All right, so some conclusions from this first part of the talk. One, it was relatively easy to find non-fixing bradyrhizobium in these natural populations. They're relatively common, and hosts are encountering them in natural populations. We inferred at least 4 to 6 independent origins of this uncooperative state within this metapopulation. And finally, in all cases, those non-fixing strains were very closely related to nitrogen-fixing relatives, suggesting that these reversals or these evolutionary losses of the cooperative trait are occurring relatively recently, potentially within ecological time. All right, so we can see this first part of the talk as kind of a primer to give ourselves an idea of the context in the system in which the host must maintain quality control. Given that the host is encountering a mix of symbionts, ranging from beneficial to these ones that we can categorize as non-fixing, how does the host respond to this range of cooperation and exploitation? All right. So one of the first things we want to do is just create a simple model that we can test. And in this model, we can imagine a simple host legume that's nodulated by multiple strains. We can infer that these are likely to be genetically different from each other based on our previous data. And we know that hosts commonly are going to be encountering strains that vary in terms of how much nitrogen they're fixing for the host. So let's imagine a model host that's being infected by a fixing and a non-fixing strain as shown here. Now, we want to test a dummy model, we'll call it a null model. In this null model, the host doesn't differentiate. It invests equally in a nitrogen fixing versus a non-fixing strain. So we want to test this model and see if we can reject it. In other words, test whether the host can differentiate these different levels of services within these different symbiotic organs. We tested this hypothesis in various ways, but rather than starting in our natural system, we started in a related model system. Lotus japonicus is actually a close relative of our wild study species, Acmaspon strigosus. They used to be in the same genus. Acmaspon was recently separated from Lotus as the New World version of the Lotus genus. We took Lotus japonicus and we infected it with its wild type symbiont described as MAFS. And we also infected it with two nearly isogenic mutants. One is a mutant in which one nitrogen fixation gene has been knocked out. And another is a mutant in which a nitrogen fixation regulation gene has been knocked out that causes the strain to fix nitrogen at a medium level. So here's a graph of growth of hosts inoculated with these three strains over time. All right, so the green strain is the nitrogen fixing strain. The hosts are growing over time. The pink strain is the non-fixing strain. The hosts are not growing because they're not getting any nitrogen, and we grow them under sterile conditions in nitrogen-free medium. And then we've got the medium strain in between that shows the host has some medium level of benefit. What we want to do is test hypotheses about host response. And we'll start with a really simple data set. That's simply looking at mean individual nodule size. So this is gauging how much investment the host is giving into each of these symbiotic organs. What we find is that the host produces a very elegant response, a geometric and proportional response in which the host forms the largest nodules when they are being infected by highly beneficial rhizobia, very small nodules when those nodules are housing non-fixing strains, and then in between for the medium strain. And we can see these at data for a 6-week and for an independent 8-week experiment. The next thing we want to do is look at the fitness effects of this response on the rhizobia. And to do this, what we can do is quantify the population size of rhizobia in each of those nodules. Those rhizobia populate the nodules, they proliferate within a growing, developing nodule, and then ultimately they're going to be released back into the soil. What we found is that this proportional response by the host is reflected in terms of the fitness effects on the rhizobia. So rhizobia that fix a lot of nitrogen, reach high population sizes, and then those medium and low fixing and non-fixing rhizobia, they have greatly reduced fitness. All right, so that's a model system. And briefly, I want to review the data when we studied the same kind of experiment in a natural system. Now, rather than nearly isogenic strains, we have strains that are quite diverse, but they vary in the phenotype of how much nitrogen they're fixing for hosts. Here are 4 different strains that we tested, ranging from non-fixing to highly fixing. We gathered the same kind of data with a series of infection experiments. And the upshot of this is that we got the same key response. When we look at the relative growth effect of each strain on a host by comparing an inoculated host versus a control host that received no inoculation, the growth that the host experienced from those rhizobia translated directly into the individual nodule size produced on those hosts. So hosts are rewarding the rhizobia in a proportional way. Now, you might think I could stop here. The hosts have a nice quality control system in order to regulate the rhizobia, and they're punishing the rhizobia if they're not cooperative. But it turns out that All these experiments are relatively simplistic, and they don't reflect the complexities of the biology in the system. All the experiments I've shown you so far have been based on plants grown under sterile conditions and only given a single strain to respond to. But of course, as I described in the first part of this talk, these plants are encountering many different diverse strains, and indeed, nodules can be infected by multiple strains. Now, there's a lot of theory in the system, and one theory about this, it predicts that the host should not be able to regulate rhizobia cooperation if nodules harbor multiple rhizobia strains. This model is based on the idea that the hosts are responding to individual nodules and rewarding or punishing them with some whole nodule mechanism. So if an individual nodule contained multiple strains of rhizobia, then the host would not be able to respond. Now, we gathered some data along these lines, and we found two interesting things. We found that nodules can actually often be co-infected. We've even found 3 different rhizobial strains in nodules when we gather them from nature. And also, we found that this evidence for a reward and punishment system worked very well even when hosts were infected by many different strains, and indeed, even when nodules could be co-infected. So we wanted to try to understand the quality control at the cellular level. To do that, we started doing some inoculation experiments that we coupled with some histological analysis of the nodules. So let me walk you through some of the responses. Here is a panel of nodules. On the top row are nodules that are infected with a highly beneficial strain, and the bottom row is one of these non-fixing strains. And this is in the wild plant Acmosphon stridiosus, and I'll show you some data from Lotus japonicus a little bit later. In the top panel, we see a much larger nodule indicative of a more cooperative rhizobia fixing nitrogen, and that's reflected in some of the key histological characteristics of that nodule. The infected region, stained in blue by toluidin blue, those are all the cells that are infected. We see it's interspersed with uninfected cells. Those cells themselves are large, and they appear to be healthy plant cells. Many of them have a large vacuole that you can see, and that's surrounded by a tonoplast. That's maintaining the structural integrity of that vacuole that harbors, of course, water and carbohydrates. We don't see any of these features in the non-fixing strain below. Well, one, we see that the infected region appears to be greatly compressed. And if we zoom into those infected cells, we can see that many of them harbor some key features of plant cells that are undergoing senescence. In all cases, we don't see a tonoplast, but instead we see that that tonoplast must have ruptured, or perhaps it never developed, because we don't see any intact vacuoles. So we see senescence that's occurring only in the infected region of the plant nodule. The other cells appear to be normal and healthy. We can zoom in and look at some of the same features using transmission electron microscopy. Again, top row versus bottom row, nitrogen fixing versus non-fixing. In the upper left corner, the TEM shows some very interesting patterns. Those dark blobs within the cell are actually bacteroids. These are rhizobia that have undergone a differentiation process to be nitrogen fixing symbionts, and they bear a lot of features similar to organelles. When the rhizobia enter a plant cell, they actually wrap themselves with a layer of plant membrane. And we call this the parabacteroid membrane. And this protects themselves from the cytosol and allows them to gain a higher level of self-regulation. Within the symbiosome, the rhizobia fix nitrogen, and we can see that these symbiosomes are maintained in the nitrogen fixing nodule. In the nodule infected with a non-fixing strain, these symbiosomes, or these parabacteroid membranes, have ruptured. They have released the rhizobia into the cytoplasm. And indeed, we can see that the counts, or the population size of those rhizobia, is greatly reduced by this process. All right, that's all what's happening within individually infected nodules. Now, the next step is to start co-infecting nodules. So here are 2 nodules with different features, a nitrogen fixing strain on the left and a non-fixing strain on the right. And now we're going to start producing plants that are co-infected at very high concentrations to try to encourage the development of nodules that are co-infected. When we started conducting these experiments, we saw some very interesting features. In particular, we found nodules that we described as sectored nodules. These are nodules that have features of both a nitrogen fixing and a non-fixing strain. The features of the non-fixing strain can be shown with the dotted line around it in this picture, where you can see that a subset of the infected cells appear to be undergoing a rapid senescence, as I described before. We only observed sectored nodules when we infected cells with a nitrogen fixing and with a non-fixing strain. And what's interesting is that the cells undergoing senescence are always localized to one portion of the nodule. We started looking at the literature and we realized we had not discovered sectored nodules, but they had been described many different times before, again, in experiments in which plants were infected with multiple strains. Here's the first example we could find. It's a soybean nodule from a paper by Hahn and Studer in 1986, in which an individual plant has been infected by a nitrogen fixing strain and a mutant that was derived under antibiotic resistance culture. All right. We can also see very nice evidence of sectoring at the transmission electron microscopy level. And we can show the same features that I described before, with subsets of the nodules harboring healthy bacteroids within intact symbiosomes, and other parts where there are cells where those symbiosomes have ruptured, releasing the rhizobia into the cytoplasm, and thus leading to a great reduction in rhizobial population size. All right, so that's all work in Akmaspan, and like you may have noticed, we don't have any markers to actually assure that these nodule cells that are undergoing senescence are actually infected with the non-fixing rhizobia. So we wanted to close that loop by going back to Lotus japonicus and using cell lines that we had marked with green fluorescence protein in the case of a fixed plus strain, and red fluorescence protein, in the case of a fixed plus strain, and green fluorescence protein in the case of a fixed minus strain. And again, these are the same nearly isogenic strains that I described before. So we co-infected plants, and we again it was relatively easy to recover nodules that had the same sectored appearance. Here we can prove that it's occurring because we've got GFP and RFP expressing plant cells that are infected with these different genotypes of rhizobia. This is very early in infection, before we'd expect the senescence to occur. So there's the nitrogen fixing strains in red and the non-fixing strains in green. All right, so here's later on during the process, and what we can see is the same sectored appearance. A subset of plant cells are infected with a fixed minus strain, but in all cases, those cells infected with the fixed minus strain are rapidly undergoing senescence, and indeed, they're even releasing some of those fixed minus rhizobia into intracellular space as those cells are rupturing. Okay. All right, so some conclusions from this part about quality control. One is this punishment mechanism, which is widely described as sanctions, it appears to be induced by plants by some kind of rapid or accelerated senescence process. And this is occurring within nodules that harbor non-fixing strains. Two, the senescence process is fascinating because it bears a lot of features of programmed cell death that has been described in plant cells. In particular, it is initiated by the rupture of the tonoplast, and then the rest of the cell starts to undergo senescence. From the evolutionary perspective, this is most interesting to me in that this process appears to be cell autonomous. And this gives this plant quality control over symbionts at a cellular level, and allows them to punish these cheaters even when a nodule is harboring multiple genotypes. All right, so I'd like to recognize the students that did all the work that I presented today, in particular, John Regas and Cami Wedlin, both of which have recently graduated. And I'd like to thank my funders and thank you for your attention.